This is the first Alzheimer's patient, a woman named Auguste Terre. It's a really touching and sad story. She came to him when she was 51 years old. She'd become paranoid. She didn't trust her husband anymore. Yeah, never mind. She was losing her memory. She couldn't identify objects. Alzheimer interviewed her many times and kept very careful records. You can look them up online of the conversations, the stilted, difficult conversations that they had. Auguste Terre died 5 years later at the age of 56. Alzheimer had then moved from Frankfurt to Munich, but he had her preserved brain sent to him, and he carefully sliced it, thin sliced it, put it under the microscope, and this is what he saw. Had this been a normal slice of brain tissue, here's what you would not have seen. You wouldn't see these big, dark blobs that are scattered around. Aggregates of protein deposits in the brain that should not be there. He called them plaques. There were also, and it's slightly more difficult to see, objects like this and these sort of flame-shaped black objects. Again, protein deposits. They're called tangles. And I want to describe both plaques and tangles a little bit because even though this was 1906, plaques and tangles are still the diagnostic feature of Alzheimer's today. So this is an image of a plaque. It's a little bit indistinct, but I think that what you can see is that it's not a simple blob. It's an aggregate of different kinds of things, proteins, broken up brain cells, and actually quite complex. These accumulate outside the brain cells in the brain. This is a normal cell, and what I want to point out to you to get to the idea of the tangles is that brain cells especially have a very elaborate inner skeletal network that helps the cell hold its shape. When that network starts to collapse in a brain cell, it can turn right in on itself, and you get something like that labeled tangle on the right-hand side. So two things are happening in the Alzheimer's brain at the same time, the deposit of plaques and the deposits of tangles. So why was he so crucial? He showed that together with dementia, the brain accumulated plaques, the brain accumulated tangles, and the brain itself was shrunken and had lost tissue. I can't tell you how many people come to me and say, my maternal grandmother had Alzheimer's disease, am I going to get it? It's not a stupid question, but you have to take great care in answering it because the likely answer is your chances are no different than anybody's. Here's why. Auguste Terre was 51 years old, died when she was 56. She had what's called early onset Alzheimer's disease. It's very rare. It represents something like 1% of all Alzheimer's. And in fact, if you look at all of the cases of people who develop it in their early 50s, not all of them are even genetic. So there's early onset, but then there's early onset familial Alzheimer's, which can be genetic, is often genetic, and there are 3 genes that have been identified as playing a role. And in this early onset form, they don't just play a role. If you get the gene, you will get the disease. It is absolutely definitive. But they may end up playing an important role in discovering new drugs, and here's why. There's an extended family in the country of Colombia, some 1,500, through whom the presenilin gene is passing. So there's a significant number of people in this large extended family who get the gene, start to show symptoms of Alzheimer's in their mid-40s, and usually die in their early 50s. They've been selected to study the effects of a drug on the basis that maybe the reason, and I'll get to drugs in a second, but maybe the reason we don't really have very effective drugs yet is because we don't give them early enough in the development of the disease. Whereas here we have a population that if they've got the gene, and it's easy to test for it, we know they'll get the disease, and we know that they'll start to show symptoms at, say, age 45 or even in and around there. So a program has started where people who have the gene in this extended Colombian family are getting drugs, anti-plaque drugs, when they're 30 years old. They have no symptoms at that point, but it's known that they will develop Alzheimer's. So this is a crucial, critical test of this one particular drug, giving it to 500 people who have the gene, and therefore we know they'll get the disease. 500 people have the gene who don't get the drug, because you need controls. 500 people who don't have the gene and don't get the drug. This program just started last year. It's going to be fascinating to see if the drug actually prevents the disease. But there's a timeline that I want to call your attention to. They're getting the drug when they're 30. They're not normally going to show symptoms till they're 45. So that's 15 years from now. And then really they don't die until they're in their early 50s. So let's say the complete experiment won't be completed for 25 years. That's a very long timeline when the world is crying out for an effective drug. And governments around the world are making, many health ministers are making statements like, by 2025, we want to have an effective Alzheimer's drug. If the Colombian experiment is the critical experiment that's going to help us determine that, say, giving drugs early is going to work, we're not going to know the full results of that for 25 years. So there is definitely a time crunch happening. What about the rest of the Alzheimer's population, the 99% who don't get it early but get it late, from 65 on up? Current estimates are that the risk at age 65, it's almost 1 in 10. By the time you get to 85, it might be 1 in 3 or even 1 in 2. What are the genetics of that? Because those are the genetics that we all have to worry about. My mother, do I worry? She got Alzheimer's late in life, in her mid to late 80s. In this case, there's only one gene so far identified that comes in 3 varieties. It's called ApoE. That's the technical name of the gene. And the 3 varieties are, with the usual clever scientific naming, ApoE2, ApoE3, and ApoE4. If you get ApoE2, 1 or 2 of them, you're actually better off than the rest of us. It actually lowers the risk. If you, like most people, 75% of the population have ApoE3, double dose, it's normal. You're running a normal risk. If you get 1 copy of ApoE4, especially if you're a woman, your risk rises, maybe as much as 4-fold. If you get both copies, 1 for your mother, 1 for your father, of ApoE4, your risk can be raised as much as 10-fold or even 15-fold in some studies. But there are puzzles even here. So you'd think, okay, man, if I get a double dose of ApoE4, I'm doomed. But half the people who have a double dose of ApoE4 never get demented. And many people who have neither 1 nor 2 copies of ApoE4 still get demented. So you see, it's different from the early onset. That early onset thing, you get the gene, you get it. If you don't have the gene, you won't get it. Late onset is a different animal entirely. And so you can't think of the late onset genes as being dictatorial of whether you'll get it or not. All they do is slide your risk a little bit up or a little bit down. So then, that does raise interesting questions about, do I want to get my genome done? Do I want to find out if I have ApoE4? Well, I've gone from thinking, yeah, I might want to know that, or maybe I don't dare know that, to I don't really care. Because what it does not say is that I will or that I will not get Alzheimer's disease. So the short answer to, am I going to get it? What are the chances of my getting it? Are, if one of your parents had early onset and you get your genes checked and you have one of those 3 genes that cause that, you are going to get it. As far as late onset, the jury is completely out on the significance. Even if you have both ApoE4 genes, it doesn't mean you're going to get it, doesn't say when you're going to get it. 678 sisters of the Catholic Order, the School Sisters of Notre Dame, were recruited by a scientist at the University of Kentucky to participate in a long-term study of Alzheimer's. They were all 75 or older. They agreed to 2 things: regular psychological and cognitive testing, and donating their brains for autopsy when they died, not before that. So my favorite character in this study was one of the very first nuns to volunteer, Sister Mary. She was tiny, and some of her fellow sisters described her as bossy. She was 80 pounds when she died. She had a tiny brain, 800 or 900 grams. Average is 1,500. 4 months before she died, she had her last psychological tests, and she scored better than most of her 80 to 85-year-old sisters. So 101 years old, she was cognitively intact. At autopsy, sliced her brain, riddled with plaques and tangles. She isn't the only case, and it wasn't even the first case of this, but was because of the focus on the nun study caused a sensation. So how is this possible? If plaques and tangles cause the disease, how then can a woman with a brain that's already on the small end of the spectrum, how can she resist the pathological impact of plaques and tangles and still be cognitively normal? What can I do to mitigate the probability that I'm going to get dementia? And I'm going to address 2 parts. What should you have done when you were young? It's okay for you young guys in the front row, but not for the rest of us. Or, what can I do now that I'm 55, 60, 65, 70? So, what happens in youth that helps prevent Alzheimer's? First and most important, education. And the statistics are actually startling. If you quit school at grade 8, your risk is this. If you continue to grade 12, your risk is lower. If you get an undergraduate degree, your risk is lower. If you get a postgraduate degree, your risk is lower. It's amazing. But it's true, and again, nobody's really clear what it is about education, because what we're talking about is people talking and writing and thinking that somehow influences the brain in some way that enables it to resist the accumulation of plaques and tangles and disease. Bilingualism. It would probably reduce your risk by 20 to 25% if you're bilingual. Because most people that are bilingual learn it early, so the brain's still developing, but there's something about it that helps resist dementia. Then there are qualities of one's personality that I don't think you can really easily control. There was a Danish study that just showed that cynics are more prone to dementia. How can you not be a cynic? I don't know. Conscientious people. Conscientiousness is easy to measure in the psych lab. It's a questionnaire thing, basically. The more conscientious you are, the less likely you are to get dementia. So, how do you control that? Oh, I wasn't going to pay that bill today, but I'm going to. And my brain is going to be better off for it. So there's lots of things going on in youth, triggered partly by the Nun study, but then there's this evidence of education and everything else that suggests as the brain is developing, and those of you who are teenagers or who have teenagers probably already know from your own personal behavioral studies, but also should know from the science, the brain isn't really fully formed until the age of 25. And the last parts to mature are the ones that deal with suppressing risky behavior. Anyway, that's a whole other talk. So let's say you did all that you're a bit cynical, you're not terribly conscientious, you quit school a little bit too early, but now you're 55 or 60 years old and you want to know what you should do. A stat that I like to quote a lot is the Ontario Brain Institute did a combination, sort of a meta-analysis of several studies that looked at what things adults, seniors, could do. And they concluded the single most important thing you can do is walk 35 to 40 minutes a day. Not mental exercise. No, you don't have to do every Sudoku you come across. The New York Times crossword puzzle is probably good for you, but if you just weren't cynical about it and you put down the New York Times and you went for a walk, because it'll probably take you less time to do a 40-minute walk than finish the crossword puzzle, you'd be mitigating your risk of dementia more effectively. Now, this again, just as things you do in your 20s seems hard to understand how they could affect your mature brain. How is walking 40 minutes a day every day? What's that doing? Well, it's pretty clear that being overweight, or especially being obese, having type 2 diabetes, having high blood pressure, having vascular issues, cardiovascular issues of many kinds, all predispose you to Alzheimer's. So if you're even doing that minimal physical activity of getting out and walking every day, you are cutting down on the chances that those are contributing to your risk. It's such a simple thing to do, too. Honestly, you could probably park at the far end of the parking lot and gain 15 minutes right there. And it's an underappreciated thing. And people wring their hands and worry about Alzheimer's and spend way too much time thinking about their grandmother and not enough time just doing this one simple, pretty obvious thing. I think that this is a good time to sort of give you a sense of how Alzheimer's spreads. To understand where treatment research should go, you've got to understand how the disease spreads through the brain. This is a slice through the middle of the brain, back of the brain, front of the brain here. This is an area, actually two parts of the brain here, called the hippocampus, which is responsible for laying down new memories. So something happens to you today, you remember it next week. That's because your hippocampus took it in, processed it, put it into longer-term storage. There's another structure called the entorhinal cortex, which is intimately connected with the hippocampus. The most grievous damage and cell loss happens first there. I'm going to run a 30-second video which encompasses a year and a half in the development of Alzheimer's in a brain. And what you can see here at the start of this year and a half period, the areas of red and purple, there aren't many purple, are healthy parts of the brain. But this is the amount of gray matter, which essentially is a measure of the number of brain cells. As it gets down to the blue and purple, you're getting almost total loss. This was created by the Laboratory of Neuroimaging at the University of Southern California. And so I'm going to start it now. And what you'll see, so this is 30 seconds that represent a year and a half. And you can see that the devastating spread of damage, which, as I said before, you lose connections, you lose the synapses, then the cells die. And eventually, there really isn't a lot of life left in this brain. And there's how it started on the left, and there's what it looks like on the right after a year and a half. So I got to bring the plaques and tangles back into that. So if you think plaques and tangles are responsible, then wouldn't you think that it starts in the hippocampus area in the sort of middle lower part of your brain, you get all the plaques and tangles there, and then as the plaques and tangles spread, the disease spreads? No, that's not how it happens. The tangles do that. Everywhere you see the most serious damage, and as the damage spread, it tracks perfectly with the distribution and number of tangles. The plaques are nowhere near. They're scattered all over the top of your brain. And yet, the plaques are considered to be the trigger. This is what virtually all the drugs that were in clinical trials in 2014 aimed to do. They either aimed to prevent the development of plaques, flush plaques out of the brain, or break them up, because they're aggregates of small protein molecules that come together, maybe 1 million of them. You break them up, maybe they're not so destructive. So there is this puzzle though, right? If the plaques are causing the disease, why are they just in wildly remote places of the brain, not where the brain is suffering the most damage? There's no answer to that question right now. So what about the clinical trials in 2014? There's no way to put a lipstick on this pig. It was a bad year. None of them worked. And in fact, one drug that actually did what it was designed to do, reduce the plaque load in the brain, did that, and the patients continued to become more and more demented. So we're in a tight situation right now when it comes to, and by the way, that Colombian study that I talked about, the long-term study, they're using a drug there that failed in its clinical trial in 2014. But the hope is, because they're starting earlier, it might work. Because here's the theory. Maybe plaques, it's too late. Like once you see plaques, all the damage has begun, and you're not going to be able to stop it by attacking the plaques. Think of them as like refuse that's just resulted from damage. What's the point of attacking them? They're not doing the damage, they're the result of the damage. Maybe, and this is the Colombian study, and others that people hope to do, maybe if we start earlier, they'll be effective. But the bad news is that as of right now, the drug pipeline, so-called, is pretty much empty. And when you look at timelines, you want to have a drug by 2025? Absolutely you would. Are you likely to have one? Well, not unless there's some, like the way things are going now, it's not going to happen. Because it takes a long time. You've got to do cell culture studies, animal testing, early clinical trials for safety, clinical trials, several stages of those before you get a drug that can go on the market. So at the moment, I wouldn't say things look good. And like I say, that's not to say that there isn't the potential for some drug to come out of left field and turn out to be actually quite important. But I wouldn't count on it. So that's where we stand right now. Plaques are still considered by the majority of Alzheimer's scientists to be the key player. They still think that tangles somehow follow along, although, like I say, nobody can explain why tangles seem to map so perfectly onto the damage and plaques don't. Alzheimer's is the ranks 4th in the US in terms of funding, behind cancer, heart disease, and HIV/AIDS. Significantly less money. I can't really imagine why that could be allowed to continue, because if you look at the numbers, and it's, we tend to think of Canada and the US, but you look around the world, the numbers are rising dramatically in every jurisdiction, except with the exception, as I said, of those a small number of studies in Northern Europe that suggests maybe the trend could be slowing. So we're at a very pivotal point. How exactly is it expanding? More in one place than another? We don't really have effective treatments. That's the story. I do, though, think that even though it doesn't look like we're making a whole lot of progress at the moment, I still have a lot of confidence in the research that we will disentangle some of these mysteries. Studies like the Nun study provide us with new ways of thinking. We've got to be really open-minded about it. We can't get stay locked into plaques and tangles. Some researchers think that it's really all about sugar metabolism in the brain, and one researcher in the States says Alzheimer's should be called type 3 diabetes, because she sees all kinds of abnormalities of insulin and glucose metabolism in the brain. Some think inflammation is really important. There are lots of theories. It's just that now we got to get the research dollars into that and really direct our attention hard without without having too much faith in the old ideas about plaques and tangles, because in the end, they might not provide the ultimate answer.